So in this video, we are going to be looking at the dark reaction, or the light independent reactions of photosynthesis, or the Calvin cycle. Okay, there we go. And what we're basically looking at is how life is created from air. It's pretty amazing. For this, we'll make 6 boxes for our notes. We'll only fill a few of those in during this video, though. So the dark reaction is actually pretty complicated. Thankfully, we don't have to know all of these steps. We're going to look at the basics of how we get from carbon dioxide to a molecule of G3P. You can see the G3P up here. That's basically what we are interested in. All right, so now photosynthesis. We learned earlier about the light-dependent reactions. Their products are oxygen, which diffuses out of the plant through the stomata, but also ATP and NADPH. So it's the light reaction where the plant was able to convert solar energy into chemical energy, ATP and NADPH. Now it's the Calvin cycle where that energy is going to be invested into the bonds of molecules as we build the life, right? G3P, we build carbohydrates, lipids, proteins, not we, but plants. So these energy in ATP and NADPH is energy invested into the Calvin cycle, and that is why it's an anabolic pathway. All right, so here in the Calvin cycle, or the light independent reactions, carbon dioxide is required, and then with that, we'll invest the ATP and the NADPH, and then make a molecule called G3P. Now, G3P actually only has 3 carbons, so 2 G3Ps form together to make glucose. But glucose is the one that we generally talk about. All right, so the Calvin cycle, when we look at this, it is a cycle. So the beginning materials and enzymes that we use, the plant uses, will be used again over and over and over again. So the first step is going to be taking in carbon dioxide, investing ATP that was made during the light reaction, and then we're going to oxidize the electron carrier, NADPH, to form NADPH, P+. And so out comes a 3-carbon sugar. That is our G3P that the Calvin cycle makes. And then we're going to invest a few more ATP to reorganize the molecules back to the starting enzymes and starting molecules. Okay, so the Calvin cycle reduces carbon dioxide to a simple 3-carbon sugar by investing the ATP and NADPH that are made during the light reaction. Okay, so plants have to have a way to store their chemical energy made from the light reactions, but they also have to make all their own organic molecules. They make their own carbohydrates, their own lipids, their own proteins, their own nucleic acids. They are literally building life out of carbon dioxide and water. Now, carbohydrates and lipids are made of carbon, hydrogen, and oxygen. However, proteins are made of carbon, hydrogen, oxygen, and nitrogen, and then sulfur in a few. And the nucleic acid, your DNA, is carbon, hydrogen, oxygen, and then nitrogen and phosphorus. So those nitrogen and phosphorus molecules are coming from the soil. But the main mass and components of plants is going to come from carbon dioxide and water. So if a plant wants to build glucose, which is C6H12O6, how? From what? What does it have available? This is where we're going to take the carbon dioxide, or the plant is, and it's going to need to add or invest some molecules, right? So it's going to take those hydrogens off of, and electrons off of the electron carrier, NADPH. So right here, this electron carrier is in its reduced form, and at this point, this right here is oxidized. So this carbon dioxide molecule that we're starting with is going to become reduced. And when you reduce something, you add electrons and you add hydrogens to it. So here in the process of the Calvin cycle, or the dark reactions, carbon dioxide is becoming reduced to form glucose. It's investing energy in the form of electrons and hydrogens, and it's building It's an anabolic pathway where there's energy investment to build. And that energy is going to be stored in the bonds of the glucose molecule and then released when our mitochondria breaks it down. So the first step in the Calvin cycle is what's called carbon fixation. And so the Calvin cycle is going to use that NADPH from the light reaction to reduce carbon dioxide into a molecule of glucose. So the purpose of the Calvin cycle, what's happening in that? I can kind of summarize it in your box number 1. Now, how do we get from carbon dioxide to glucose? Well, CO2 has very little chemical energy, but glucose, C6H12O6, has lots of chemical energy. It's reduced, it's endergonic, and so in the process of building glucose from carbon dioxide, there's a series of steps, many uphill steps. And this is where we take that vocabulary word, anabolic pathway, into consideration. Here it's a series of steps and it's building, it's energy investment, a bunch of like endergonic reactions occurring. And each step is catalyzed by a specific enzyme. So the energy that comes from ATP and NADPH are going to be invested. So not even knowing anything about the Calvin cycle, we can think about how, okay, I'm starting with carbon dioxide and I need to end up with glucose. So I need to invest and I need to build. And we know that anabolic pathways are building pathways. So here we're going to take our carbon dioxide and we're going to invest ATP and we're going to invest NADPH, the products of the light reaction. And in the process of the Calvin cycle, we'll build G3P, has 3 carbons, another G3P, has 6 carbons. So when we look at the Calvin cycle, it's an anabolic pathway that builds with the products of the light reaction. Okay, so the Calvin cycle, here we go. We're going to start with a 5-carbon molecule. This molecule is called RuBP. That's the nickname. But here we have 5-carbon RuBP, and we have 3 of them. So here you can see there's 15 carbons that are present. So at the end of the Calvin cycle, there should be 15 carbons that we can use again. So here, we're going to take 3 molecules of carbon dioxide. I'm only showing you the carbons of the carbon dioxide molecules. Okay? So the first step of the Calvin cycle is called carbon fixation. So here I have my molecule of RuBP. Now, carbon fixation is where an enzyme called Rubisco is going to take one of the carbon dioxide molecules and attach it to RuBP. So let me show you again. Here you have an enzyme called Rubisco, and in its active site, it's going to fix a carbon dioxide to RuBP. Now we have a 6-carbon molecule. So an enzyme called Rubisco attaches carbon dioxide to RuBP. This is going to happen multiple times. Here's another RuBP. Rubisco comes and does carbon fixation, taking a carbon dioxide out of the air and attaching it to a RuBP molecule. And then we have it happen a 3rd time. So in this first phase of the Calvin cycle, we have successfully incorporated 3 molecules of carbon dioxide. We've taken it out of the gas form and physically attached it, forming 3 molecules that have 6 carbons each. Now, these carbons, now we have 18 carbons. Now, these are pretty unstable and pretty quickly they separate. We still have our 18 carbons though. All right, so in carbon fixation, the enzyme Rubisco attaches a carbon dioxide to RuBP. The carbon dioxide is fixed onto a 5-carbon molecule, forming a 6-carbon molecule that quickly splits to 6 3-carbon molecules. Okay, so go ahead and try and process the first phase of the Calvin cycle called carbon fixation and what happened, and you can summarize that in your box number 2. Okay, so in the second phase called reduction, here's what's going to happen. We're going to invest some ATP that we had made during the plants made during the light reaction. And here, oops, that's on a timer. All right, there we go. So we're going to take a phosphate off of an ATP, forming ADP. And now these 3 carbon molecules are being phosphorylated. We're investing here. So this happens 6 times. All right, so there we go. Now for simplicity, we're just looking at the carbons, even though the phosphates are still attached here. Okay, so now we invested some ATP, and our next part is, next phase is the reduction phase. And in the reduction phase, these electron carriers are going to become oxidized. I'm just going to put ox here, oxidized, which means they're going to donate electrons or donate hydrogens. And so I'm going to show you on one of them. So here you take, there's our phosphates. Okay. So here you have one electron carrier, NADPH, is going to become oxidized. And so in that process, you can see it changed the 3 carbon molecule. So we can watch this happen again. So these electron carriers are becoming oxidized, and the carbon molecules are becoming reduced. Oops. So during the reduction phase, 6 ATP and 6 NADPHs are invested during endergonic reactions. So if you remember the light reaction, you made that electron carrier, NADPH. So this is carrying the electrons and hydrogens from the light reaction over to the Calvin cycle. We also had ATP synthase making ATP, and that's where that's investing also in the Calvin cycle. Okay. So now, one of these molecules, this 3 carbon, remember the 3 carbons we took in during photosynthesis? Well, now here we're going to take this 3 carbon molecule, and this is going to be our G3P. Now, G3P is the, oops, just kidding. G3P is what's used to build all other organic molecules. If you take a G3P and a G3P, now you have a carbon. You can also take the G3P that has carbon, hydrogen, oxygen on it, and you can build lipids. You could build amino acids if you just attach another nitrogen to it. You can build DNA. So this G3P has incorporated carbon dioxide from the environment into a molecule that then can be modified in the cytoplasm by other enzymes to build all the molecules essential for life. So G3P is now the building block for plants to then build carbohydrates, lipids, proteins, and nucleic acids. Now, once G3P goes to the cytoplasm, where other enzymes will build those larger molecules, we're still left with the Calvin cycle. So this Calvin cycle, our next phase, oh, here we go. G3P is the main product of the Calvin cycle. It is the building block for all organic molecules. It is made of carbon, hydrogen, and oxygen, and it will go to the cytoplasm where it will meet its fate. Okay, so we can go ahead and kind of summarize about G3P in our box number 3. All right, so now for the regeneration phase. Here we have 15 carbons like we started with, and in this we're going to invest some ATP. And ATP will be used to reorganize and rearrange basically those carbons back into the 3 molecules of Ruby P that the Calvin cycle started with initially. And so in the regeneration phase, it's taking those 15 remaining carbons, investing ATP to reorganize them back into Ruby P, where now we can do carbon fixation all over again. So you can kind of summarize the regeneration phase in your box 4, and then see how we would start again with Ruby P and the enzyme Rubisco that would do the carbon fixation in the first phase, like that. And that is the basics to the Calvin cycle. Good job.